Good afternoon, everyone. I am Sahil Chahal, guided by Professor Parkin Tube. I am working as a PhD student in his lab and the Department of Biomedical Sciences. Today's title of my presentation is Dual Mechanism mRNA Delivery via Fluorinated Sorbitol Polyplexes, Enhancing Cellular Uptake and Endosomal Escape for COVID-19 Vaccination. The science of mRNA vaccines. First, we should basically understand how mRNA vaccines work, how we can utilize them. mRNA, as everyone is well aware, the mRNA is used to deliver the instructions for building the protein identical to part of a virus or the bacterium. Similar technology was utilized for the COVID-19 vaccine also. The spike protein that was utilized for the treatment, in which the virulent part was the removed and the remaining the coding part, that was the ORF region, which was used for generating the antibodies, so that when in the future the patients are encountered by the same virulent or the pathogen, that antibodies can fight against the corresponding virus. So that's how the COVID-19 vaccines work, and that's how the mRNA vaccines work. Nanoparticles for mRNA delivery. There are different types of nanoparticles that are widely used all over the world: lipid nanoparticles, polymeric nanoparticles, protein nanoparticles. Lipid nanoparticles, well aware, everyone is well aware. Moderna vaccine and the BioNTech, Pfizer, all the vaccines use the LNP components, which includes the ionizable lipid, helper lipid, cholesterol, PEG lipid, and mRNA. Now everyone is well aware of the advantages because we received the vaccines, but we can see there are the many patients who are suffering from the heart attack, and they have been complaining about the other side effects of the mRNA vaccines that have been received by the patients. So when the further research has been done, and it has been found that the lipid nanoparticles or LNPs, they are not safer for the delivery of the vaccines, especially when the intramuscular injections are being given. And for that, we in our lab have found out the solution. Instead of using the modern LNPs, we can utilize the polymers for the delivery of the mRNA vaccine. Question is, how polymers are better? So in one simple line I can answer is with the LNP, it can target to other organs also, but with the polymers, it stays in that localized part where the injection has been given to the patient or the any person. But for the LNPs, they bind to the apolipoprotein and target the liver, and the major expression has been observed in the liver also, which cause the liver mediated toxicity also. This was the preliminary study that was conducted in our lab, and we found that in intramuscular injection of the modern LNP, we can see the expression of the mRNA, reporter mRNA, was observed in the liver also, as well as the site specific also. But with the polymer, we can just observe the expression at the in the local part where the injection was given. Now here there was this another study that was conducted in our lab, where we are optimizing the different types of polymers for targeting the different organs, where we have found the specifically spleen targeting the polymers, liver targeting polymers. And here we can see the modern LNP, it can target almost liver and spleen equally if it is injected by the tail based route. So we have the proof of concept that the polymeric vaccines can be used to target the specific organs by the intravenous injections, as comparison to the modern LNP, which can target any organ, and we can suffer through the severe toxicity due to the LNPs. This was the paper published by our lab in the Advanced Healthcare Materials, where this is the schematic representation of that. And here we designed the low molecular weight polymeric delivery platform for the mRNA, where we use this sorbitol moiety, which is for the endosomal escape. Which is for the endosomal escape. How it enhances the endosomal escape is due to this hyperosmotic pressure that has been created, and due to the hyperosmosis, it helps the nanoparticle penetrate through the cell membrane, and then release the nanoparticle in the cytoplasm. So specifically, it enters via the caveolum mediated endocytosis. There are different other pathways also through which the nanoparticle can enter the cell membrane, but caveolum mediated endocytosis is the only pathway, or we can say, or the pathway where the maximum transfection has been observed as comparison to the other pathways. and we also did the fluorination to enhance the TLR4 activation, and we observed in our results that the immune responses were like enhanced even after the first and the second dose that was observed by us. And as comparison to the modern LNP, we found the more of the sustained expression. So this was the basic schematic representation of our work. This is the NMR that we and the characterization and the fluorination that was confirmed by us, like FTIR spectra. We can see the different peaks that are for the characterization, like the carbonyl and the other peaks. And this is the proton NMR of the we can see the sorbitol and our final polymer. Here we connected the size based assay was conducted to check the size of the nanoparticle, and we found our nanoparticle was less than 200 nanometer. And here the gel retardation assay was conducted to check the where the mRNA and the nanoparticle is binding strongly and the polymer is binding strongly, and we are getting the our polymer is effective or not. And we found that here the mRNA is not coming out of the well as comparison to our control. And this is that FE10 image. We can see the clearly the formation of the nanoparticles. Then further we did the cell uptake assay using the Cy5 mRNA. We prepared the Cy5 mRNA where we labeled the mRNA with the dye, Cy5 dye, and we tested the internalization of the mRNA in the cells using the PFS nanoparticle, and we found the significant transfection, significant cell uptake was observed. And for the cell viability of the polymer was tested, and we found that it was like almost non-toxic. And then the transfection efficiency of the polymer was tested, where it was found that the even the it was showing the expression at certain weight ratios. Then there are the different internalization pathways. As I mentioned initially, caveolum-mediated endocytosis bypasses the acidic compartments. All other compartments lead to the degradation of mRNA, while the lysosomal or the late endosome escape. So that's why the caveolum-mediated endocytosis helps in the better transfection, because it directly it kind of we can say it is provides the escape mechanism for the mRNA for the translation. Here we use the different inhibitors to prove that our mRNA or our nanoparticle enters the cell by the only the caveolum-mediated endocytosis, and the other pathways like clathrin or the different other pathways, macropinocytosis example, they don't contribute to the uptake of the nanoparticle. And that was confirmed by the FACS analysis and the transfection efficiency also. And we found that our PFS mRNA showed the better uptake as comparison to the other control nanoparticle. Then further for the sorbitol that we utilized for the internalization of polyplexes, since we are this is just the preliminary study that was it is just a proof of concept actually. We believe that our polymer enters because of this sorbitol via the COX-2 pathway. So since this pathway is not well explored all over the world, so for testing the efficacy of this pathway that our nanoparticle enters via this COX-2 channel only, we use this COX-2 inhibitor. And after using this inhibitor, we found that the internalization of our nanoparticle was significantly reduced. That was confirmed by the cell uptake as well as the transfection. Here we can see we use this inhibitor in the increased concentration, and there was the reduction in the uptake was observed, and similarly reduction in the transfection was also observed with the PFS PS we can see. But PEI we didn't observe that kind of things because sorbitol was not present with PEI. Then we tested the in vivo expression of our polyplexes in the mice. It was tested, and we found that the sustained expression of the mRNA was observed in the mice after the intramuscular injection. But we can see here Moderna LNP, in which the transfection was reduced after the certain time, but our polymer maintained the expression for the longer time, which proves the efficacy of the polymer over the modern LNPs or the lipid nanoparticles. Then we did the ELISpot analysis for for the cytokine secretion and the antibody activity, and we found that the PFS spike mRNA induces the strong IFN gamma T cell responses, and the booster dose enhanced the immunogenicity, and the elevated cytokine secretion with PFS was observed. And comparison to the LNP, we found that the our PFS platform is showing the sustained expression. It is maintaining the expression as comparison to LNP, which is showing the significant drop from the expression. Then further we did the PRNT assay also to check the neutralizing antibody titers, and we found that after booster dose PFS spike mRNA maintained the strong antibody levels through slightly lower than the LNP, and the plaque reduction assay showed LNP and the highest neutralizing activity. Post priming with PFS mRNA showed the significant effects. This is just the further exploration of our the polymeric platforms, where we are using the machine learning to identify the carriers, where we have screened almost more than 100 polymers, and we found the best carriers that are showing the maximum transfection as comparison to other polymers. And here we, after the using the machine learning, we tested in the in vitro cell viability as well as the transfection efficiency, and we found that the some of the polymers showed very strong expression in the in vivo system also and in vitro also. So out of these polymers we have the very strong multiple candidates that we will be testing in future for our further studies, and same polymers we have tested intravenously also, and we have obtained some results definitely in the future that we would be interested in sharing. So for the future directions, since as I mentioned we are currently integrating the machine learning for the design of the library of the polymers that can be utilized for the designing of the new kind of nanoparticles for the transfection as comparison to the modern LNP, and the therapeutic mRNA applications. Currently we are doing the experiments where we are using the metabolism based therapies as sparginase and the IL-15 cytokine based mRNA therapy, and the further the synergistic combination with the radiotherapy also we are currently doing the studies. And since we have expanded our research in the mRNA using our polymeric platforms. Thank you. That was my presentation. Hope I presented well. Thank you.